Thank you for the introduction, Annie. I manage Bio-Rad's team of European antibody sales specialists, and today I want to talk to you about our TrailBlazer antibodies. These are recombinant phage display-derived antibodies that feature something called a SpyTag. And today I want to describe this technology and how it can be used to overcome some of the challenges that are encountered when customers are developing PK assays. When discussing the scope and the success criteria of the project, antibody specialists such as myself will talk to our customers to really understand the full breadth of the project, because with a technology as flexible as ours, there's lots of areas of consideration that we can build into the design of the project. And I'd just like to go through a few of these typical things now that we look at. Firstly, there's the nature of the analyte that we're measuring itself. Does it exist in more than one form? Is it going to be in a particular matrix that may interfere with the antibody or the sample? What level of sensitivity is needed? What concentration is the analyte going to be present at? And are there going to be other proteins or analytes in the sample that are very similar to the target analyte, to which we may get undesirable cross-reactivity unless we really design a strategy to remove it? Secondly, we've got to think of the antibodies we're producing themselves. What format are they in? What assays are they going to be in? What role are they going to play within those assays? And then finally, we need to step back and look at the wider scope of the project. What is the study that these antibodies are going to be used in? What's the likely total volume of antibody that will be needed to last the duration of that study? And what level of characterization, quality, and QC testing will these antibodies need to go through to satisfy the requirements of the team designing the assays to ensure they have the robust assays they need for their preclinical and clinical work? With these considerations in mind, I'd like to talk about some of the core features of the Bio-Rad custom antibody service, which is based around the Hucal Platinum phage display antibody library. Firstly, I'd like to look at specificity, so how the actual biopanning methods or the library screening methods can be manipulated to generate antibodies that meet the specificity requirements of the project. Next, to look at the antibody format and how we've incorporated SpyTag technology to enable very rapid switching of antibody formats at the protein level in less than 1 hour. Then when it comes to prototyping or the testing of the antibodies that we deliver to customers, how the different antibody formats we can provide can be tested in different applications, different roles and different assays to test their suitability. And then finally, once selected antibody clones or antibodies have been selected by the customer, the sort of considerations that then become important for characterization and validation of critical reagents that will be used in the assays going forward for the study. So taking these in order, let's look at how an in vitro method of generating antibodies can be used to generate antibodies of very high specificity. So it's probably worth considering what specificity actually is. This definition is from the European Medicines Agency, which defines specificity as the ability to assess unequivocally the analyte in the presence of components which may be expected to be present. So if we have a think about that, does the antibody bind the target? And if the target exists in multiple forms, are we looking to measure one of those forms? Are we looking for a cross-specific antibody that recognizes all of the forms that the analyte can be present at? And because the analyte is probably going to be present in a complex matrix, such as human serum, does the antibody bind anything else that we could expect to be present in that matrix that might be very close in terms of homology to the target analyte? And we could even think more broadly, is there anything else in the matrix that may actually interact with the antibody and give a false signal? A lot of proteins in serum can interact with the FC domain, for example, of antibodies, such as human anti-mouse antibodies or rheumatoid factor, or if you're doing cell-based assays, FC receptors, etc. So there are a lot of things potentially in the sample that could either quench the signal or could give a false positive signal or could interact in a lot of ways. So there are a lot of considerations to really think about specificity when you're sort of really at the early stages of designing the scope of a project. The ability to guide and influence the specificity of the antibodies that we generate during our library screen really comes down to the nature of the technology we use, which is phage display. For anyone not familiar with phage display, at its most simple, it's a physical link between genotype and phenotype. So displayed upon the surface of the phage, we have an antibody fragment. It's the fragment of antigen binding, or the Fab as it's known. And then within the phage mid, or the phage genome, we have the gene encoding that particular antibody fragment. What in practice this allows us to do is take the customer's protein of interest, immobilize it. We can put our library of phage, which display different antibody Fab fragments upon their surface, against the protein. Any phage displaying antibody fragments that are specific will bind and be immobilized themselves. The non-binders are washed away. The binders are recovered, and then we're able to sequence the gene. So essentially then we can recover the gene sequence of the antibody Fab fragment that recognizes the customer's protein of interest. Like any in vitro process, we have a lot of flexibility to manipulate the conditions of the process to achieve a particular outcome, say compared to a classical method of generating antibodies using, say, animal immunization. One of the tactics we use is called the blocking strategy, and I'll give you an example in this slide. So in this scenario, we have a target antigen shown in blue, so say this is a human protein. But we also have a closely related antigen that we don't want our antibodies to cross-react to. So say this is a non-human primate homologue of the human protein that's 95% conserved by sequence, say. So in this scenario, what we would do is we would immobilize the target antigen. We'd use it as bait, as shown in the diagram. We'd coat it to a solid surface. But in the solution phase, we'd use a higher concentration of the closely related antigen. So in this case, the primate homologue. And essentially, because the closely related antigen is present at a higher concentration, it should compete with the immobilized target antigen for any phage that is displaying antibodies that are cross-reactive. And because it's present at a higher concentration, it should win that competition. And the closely related antigen is present in the solution phase. It's not immobilized. So when the wash step comes along, the closely related antigen and any phage displaying cross-reactive antibody fragments are lost in that wash step. So if we apply this for all 3 rounds of the biopanning enrichment process we use, what we should end up with is only phage that display antibodies that recognize features that are uniquely present on the target antigen, but are not present on the closely related antigen. The generation of anti-idiotypic tool antibodies, in this diagram shown in purple, to therapeutic drug antibodies shown in gold, is a good bioanalytical relevant example of how we can apply our library screening capabilities to generate specific reagents. Not only are drug antibodies a common class of analytes that we get asked to generate tool antibodies to, but if you think back to my earlier slide where I talked about specificity, they're an example of an analyte that can exist in potentially more than one different form in your matrix. So for example, the drug antibody can often exist in the free form, but if the target of the drug antibody is present in the serum, for example, if it's a receptor that is shed into the serum, or if it's a serum protein, your analyte, your therapeutic drug antibody, can exist in complex with its target, and so won't be present in the free form necessarily, or not entirely present in the free form. Also, if you think about therapeutic antibodies, they're also present in a background of highly similar proteins. In this case, the therapeutic drug antibody is present in a background that contains a much higher concentration of a patient's own antibodies. So you're trying to discriminate between a drug antibody and a human antibody background. So it's quite a good example of an analyte with lots of specificity considerations to take into account when going into the study design. So with our technology, not only are we able to generate anti-idiotypic antibodies, but we're actually able to use different strategies to generate different classes of anti-idiotypic antibodies. The most common we get asked for is the type 1 anti-idiotype. So in this scenario, we're generating antibodies that recognize the binding site or the paratope of the drug antibody. These are useful to the customer because they allow the customer to measure the free form of the drug in serum, and also type 1 anti-idiotypic antibodies are usually neutralizing, they're usually inhibitory, they bind to the binding site of the drug antibody, and in turn this prevents the drug antibody binding to its target. Our strategy for generating type 1 anti-idiotypes is simply to immobilize the drug antibody, to block with an isotype control, so this would be a similar framework, similar germline, subclass, IgG, kappa, lambda, isotype control to the drug, and also with human serum. So this really directs the specificity towards that area that's most different, which is the paratope. For customers who know that the target of their drug is going to be present in the serum as well as the drug itself, they often require a type 2 anti-idiotype. So this is an anti-idiotype which will recognize a unique feature outside of the paratope of the drug antibody. What this will allow the customer to do is measure the total drug regardless of whether that drug is in the free form or in the complex form with its own drug target. To generate type 2 anti-idiotypes, we use what we call an alternating approach. So we will enrich upon the free form of the drug. We will also enrich upon a complex form of the drug. And we form the complex by coating a solid surface with the drug target at a high concentration. Then we apply the drug at a lower concentration so that we're confident that all of the paratopes are occupied. So by alternatingly enriching between the free and the complex form, we should generate antibodies that recognize features that are outside of the paratope, but then by blocking with the isotype control in human serum, we ensure that those features outside of the paratope have to be unique to the drug. They can't be common antibody framework features. The final type of anti-idiotype we generate is the type 3 anti-idiotype. So this is a complex specific antibody. So this will only recognize the complex form of the drug, but not the free form of the drug. To generate a type 3 anti-idiotype, what we will do is we will build the complex and we will use that for all 3 rounds of enrichment, but we will alternatingly block our library between the different components of the complex. Therefore, we'll generate antibodies that don't recognize either component of the complex on its own, but they recognize that unique feature that is present when the complex is formed. So between the type 1, type 2, and type 3 anti-idiotype, we try to provide our customers with an entire toolbox of antibodies to measure the free, the total, and the bound form of the drug. In this slide, I've also included another example on the far right. So this one that says other is actually a diagram of etanercept. And etanercept, for anyone not familiar with it, is an Fc fusion protein. So what you have is a TNF-alpha receptor fused to a human IgG-Fc domain. Now this is quite a challenging target to generate antibodies to that uniquely recognize this, because human Fc is obviously present in the serum because it's present on human antibodies. But the TNF-alpha receptor used in etanercept has a soluble form which is present in the serum, and it was the soluble form that they used when they made etanercept as a drug. So both the Fc component and the TNF-alpha receptor component are going to be present in your matrix. So how do you generate an antibody that recognizes the receptive Fc fusion protein? And the answer to that was to target the unique feature of the drug, which was the junction between the TNF-alpha receptor and the Fc domain. This is a sort of one feature that's probably not going to be present in your matrix because it's not present in nature. So this is quite an elegant example of how you can use, library screening strategies to generate uniquely present antigens that then allow you to measure your drug without cross-reacting to the very similar analytes in the background. And one thing I'd like to point out about all 4 of these different strategies is none of them use peptides. So quite often we hear customers, they assume that they've identified by sequence analysis something that's unique on their drug, so they assume they need to make the peptide, we generate antibodies to the peptide, and then we see if it binds the conformational folded form of the protein. We can take this approach, but our preferred method of working is, as you've seen in these diagrams, we work with the whole proteins in all cases, the whole drug antibody or the whole Fc receptor fusion, and we use high stringency blocking to remove all of the antibodies to the sort of common or conserved domains, and really focusing on those uniquely or those unique features that are present on the target antigen but not present in the background proteins. So yeah, I hope that's given you some good examples of how we can use our screening strategies to generate these highly specific antibodies. And I'd like to give you another example in the next slide as well, going beyond just binding for, say, a ligand binding assay, but actually to look at generating something that's specific and functional. Neutralizing antibodies are something we get asked for quite a lot, less so for PK, more so for your sort of anti-drug antibody assays, immunogenicity assays, or neutralizing antibody assays. But quite a lot of biologics are enzymes or have some kind of active site or binding site function that customers want neutralizing antibodies or inhibitory antibodies too. This particular example I chose is an academic publication because a lot of the work in terms of neutralizing antibodies we've done for customers in the area of biologics is proprietary, so I don't have the freedom to share those stories. But this tells the story quite nicely. So in this example, we were looking at generating neutralizing antibodies to a West Nile virus protease. And what the customers were able to provide us with was the wild-type protease, but also a version of the protease with a single amino acid substitution in the active site, which was non-functional. So if you think back to the diagram I showed of the blocking strategy, what this allowed us to do was to immobilize the wild-type protease and use this as bait for our enrichment process, and then to use the single amino acids knockout version of the protease at a higher concentration as our closely related antigen or our blocking antigen. And essentially what this achieved was it blocked out all of the antibodies to all of the common features, i.e. the features that were present on the closely related antigen, which were essentially every feature outside of the active site. So it focused all of the selectivity of our enrichment process into that active site. So this was essentially a very, very high stringency block because there was only one amino acid difference between our bait protein and our blocking protein. And so we only got 3 antibodies out of this library screen, as shown in the diagram. And what you see in the top right is typically the data we produce when we do a library screen. So in the blue bars, you see binding on the target version of the protein, the wild type, and in purple, what you see is binding on the one that's one amino acid different and non-functional, and then some sort of essentially diagram showing that there's no binding on unrelated proteins. So as I said, we only got 3 hits, but if you look at how the customer then used these, they use them in protease activity assays. So what you see on the y-axis is protease activity, and then on the x-axis you have increasing concentrations of our antibodies. So what you see is, although we only recovered 3 antibodies from this library screen, all 3 were neutralizing. So we really designed the specificity and the function of these antibodies into the generation process, and with the high stringency blocking, it seriously limited our output, but the output met the success criteria. So if you contrast this to maybe if you tried this in a mouse hybridoma model, in reality you'd have probably immunized the mouse with the wild-type protease, and then you'd have had to screen through a very large number of clones hoping to find one that was active site specific. Whereas our approach was to actually stringently block everything away apart from those antibodies that recognize the active site. Okay, so let's assume we've generated antibodies that meet the specificity requirements. Now, another area of consideration is what format do you want to receive those antibodies in order that you can evaluate them in the various applications that you have in mind. And this is an area where we used to have choice, and we've now introduced flexibility using a technology called the SpyTag technology, which I will explain. Historically, our customer's choice was essentially limited to a list of destination vectors we had. So the customer would choose from a list the format of the antibody they wanted at the start of the project. After we'd done our enrichment work in the phage display, we would subclone into a destination vector that would produce the antibody in that format. And if the customer wanted to change the antibody format, we would have to subclone into another destination vector and then do a test production. And this was a process that was quite expensive. It would take 4 to 6 weeks. And it wasn't something that lent itself to testing sort of 10 or 15 or 20 clones in multiple different formats. What we have now done is we have added a new format which features something called a SpyTag, and this allows us to essentially make our antibodies in one format and then at the protein level to rapidly convert that. And I will go into some more detail about this in the next few slides. Okay, so what is SpyTag? SpyTag gets its name from strep pyogenes, and within strep pyogenes is a fibronectin binding protein. Within the fibronectin binding protein there's a collagen adhesion domain, and within the collagen adhesion domain there's an intramolecular bond between a lysine at position 31 and an aspartic acid at position 117. And this bond forms spontaneously with the reaction energy from a glutamic acid at position 77. The academic group who developed this technology essentially showed that if you produce a 13 amino acid tag and a 15 kilodalton components of the domain separately and then recombine them, this intramolecular isopeptide bond forms again. So this bond, as I mentioned, is a covalent bond, it's an amide bond, it's an irreversible bond. And I will show you how we've used that in the next slide. At Bio-Rad, we've taken the SpyTag technology and we've applied it to antibodies. Specifically, what we do is we now make most of our Fab antibodies featuring the SpyTag on the C term of the heavy chain of the Fab, in between a flag and a his tag. The his tag is what we tend to use for purification. And we also have a menu of pre-prepared catcher domains. So these could be single catcher domains, either labeled or with extra tags on. It could be 2 catcher domains linked together to form a dimerization domain, or it could be mammalian expressed FC domains, either of human antibodies or other mammalian species FC domains, that also feature the catchers. And what this enables us to do essentially is to make the Fabs and then at the protein level to ligate them to potentially multiple different formats, so that we can, say if we do a custom antibody generation project for a customer, we produce 10 unique clones. The customer can elect to receive those clones all in the monovalent format and all in a bivalent format, maybe with HRP, and all in a fully human IgG1 or IgG4 or rabbit immunoglobulin format. And really what this allows the customer to do is try multiple antibodies in multiple different formats in multiple different assays. So when they're going through all the antibodies we've generated, they're not limited to trying them all in one particular format, but they can try them in all different roles that they have in mind for them. In addition, we're not just using this technology so that we can produce the antibodies in multiple formats for the customer. We're also making these catch domains available as catalog products on our website, so the customers can do these reactions themselves. The customer can simply buy a monovalent Fab of those featuring the format, and they can buy the catches themselves. These reactions are spontaneous. It takes about an hour, and it's a very robust reaction. It can occur in most pHs, salt concentrations, etc. So really, it's going to give the customers flexibility to at any time make multiple different formats themselves according to their needs. So we see a great potential for this technology, and the catalog antibodies you can see on this slide are probably just a start. This is a menu that we plan and intend to expand to give customers more and more options, more and more flexibility. In deciding which antibody formats the customer may wish to receive the antibodies in, some consideration has to be given to the assays that those antibodies will be used in and the roles those antibodies will play. So in the next few slides, I'd like to give some examples of typical assays, either that we've worked up ourselves or that we know customers use our antibodies in. For pharmacokinetic assays, we're often asked as an additional service to do sandwich pair screening. That's regardless of whether the analyte is a drug antibody or a or a different type of protein such as an enzyme. And this is something we can typically do. We can look at all of the antibodies we've produced against a particular target, and then we can look at the pairings of those antibodies and see which give the best sensitivity in a titration curve. One consideration when generating PK assays for therapeutic drug antibodies, in this diagram shown in gold, is that it can be a good idea to capture your therapeutic drug antibody with a monovalent Fab format, because the monovalent Fab format can be coated thinly enough so that in theory it should be spaced far enough apart that only one arm of the therapeutic drug antibody is captured. This should leave the other arm of the therapeutic drug antibody free for detection, which allows the customer to achieve specific capture and specific detection, which can give lower background than, say, specific capture with an anti-FC detection. Another thing this diagram shows is a comparison between both our the different SpyTag formats of the Fab antibody versus a classical mammalian expressed IgG version of one of our recombinant antibodies that was generated through cloning into a mammalian expression vector. And essentially what you see is the SpyTag versions give very good equivalence compared to the sort of classical full IgG version that we used to make. The increasing popularity of antibody fragments or nanobodies or essentially small antibody-based binding molecules provides some challenges and also some interesting opportunities when it comes to PK assay development and generating reagents to use in those kind of assays. So I thought I would include a slide on some work we did generating anti-idiotypes to the drug Ranibizumab. So Ranibizumab is a 48 kilodalton Fab fragment rather than an intact IgG, and it's specific to vascular endothelial growth factor A. Because it's just a single Fab essentially, we can't use the type of bridging PK format that you saw in the previous slide because there's only one binding arm. But what we were able to do was generate both type 2 and type 3 anti-idiotypes to Ranibizumab. And using these in combination with antigen capture, we were able to essentially generate titration curves that would enable the measurement of Ranibizumab in, I guess, ocular fluid in this case. So essentially what we did in both of these assay formats, the principle is very similar. The vascular endothelial growth factor A, which is the drug target, is coated onto a solid surface. Ranibizumab, shown in gold, is allowed to bind. And then either our type 3 anti-idiotype, our complex specific one, as shown in the top diagram, is allowed to bind that complex, or our anti-total drug, our type 2 anti-idiotype, is allowed to bind. And these will be labeled with some kind of reporter, which would then allow the development of the titration curve. So I thought that was quite an interesting example. Whilst pharmacokinetic assays have been a theme in this presentation, we know that a lot of the reagents we generate as capture or detection reagents for PK assays are also used as positive controls in anti-drug antibody assays. So I wanted to include a slide on how anti-idiotypes, again shown in purple or blue in this diagram, can be used as positive controls or serology controls in these kind of assays. So in these diagrams, the assay principle is that you have a mobilized drug shown in gold, regardless of whether it's an antibody or a different type of protein. And you also have labeled drug, and a bivalent format of an anti-drug antibody forms a bridge between the immobilized and the labeled drug, which enables the generation of a signal. Also shown in this slide is essentially how the SpyTag technology can be used to prototype different antibodies for these type of ligand binding assays before maybe a final decision is made on a particular number of clones, which are then converted to full IgG if the customer wants to have something, I suppose, as representative to the human immune response as they possibly can get, which would be a full IgG rather than a SpyTagged format of the IgG. And this diagram is quite nice because what it shows is essentially that we generated 3 different antibodies of differing affinities. On the left-hand side, you see them in the SpyTagged format, and on the right-hand side in the mammalian expressed full IgG format. And what you essentially see is the equivalence is very good. The lower affinity antibody was lower affinity both in the SpyTag and the mammalian expressed version. The medium was medium and the high was high. So that that trend of affinities did not shift. So it showed that the SpyTag format is fairly representative of what you will get if you then take those antibodies onto full mammalian expression. On the subject of affinity, it's also worth mentioning one of the benefits of SpyTag is that we can work with the antibodies all the way through the generation in the monovalent format. So we get really nice, clean affinity signals if we're doing things like K-off ranking or affinity measurement. But one of the downsides of working with monovalent formats, although you get nice, clean affinity measurements and you can select highest affinity binders, the monovalent format itself is a low avidity format. So what it allows us to do is select the highest affinity binders and then rapidly convert them to the higher avidity bivalent format immediately before sending them to the customer. So for those customers where affinity is critically important, where their analyte may be present at maybe low concentrations and a high analytical sensitivity is required, the SpyTag gives us the option of doing what would have been a two-step, more expensive, more time-consuming process, and it allows us to condense that into a single process. The Bio-Rad custom antibody service is not limited to just generating antibodies for bioanalytical reagents. We do a lot of work with academics, making antibodies for fundamental research. We do work with early discovery departments of biopharmaceutical companies who make antibodies to contaminants. So we have a lot of capabilities and a lot of ways of validating the antibodies both during the generation process and afterwards. So, for example, we can screen antibodies by Western blot flow cytometry. We can use them in bead-based assays. We have a lot of capabilities, so please don't limit your thinking on what your antibody supplier can do to just maybe simple ELISAs and ligand binding assays. If you have particular requirements, it's good for us to know early on because we can actually build that into the design of the library screen to maximize the chance that we get antibodies that will work best in the applications the customer is using. So let's assume that we've generated antibodies that meet a customer's specificity requirement. Consideration has been given to the roles that those antibodies will play in the various kits, and appropriate formats have been chosen, and the antibodies have been sent to the customer. That evaluation has taken place, suitable clones have been identified, and we're now approaching the stage where the assays will be made. The customer needs to think about quantities, they need to think about validation. And this is, again, an area where we work with a lot of IVD manufacturers. Again, they have very similar considerations in terms of validating their kits, continuity of supply, lot-to-lot reproducibility, etc. So we're familiar with working with these kind of environments. So let's just look at a few considerations and capabilities that we have at Bio-Rad to support that kind of activity. Recombinant antibodies are, by their nature, quite well-defined reagents. The DNA sequence of the antibody is known, and lot-to-lot reproducibility should be good because reproduction of the antibody is simply made by transfecting either E. coli or mammalian cell lines with an expression vector. So they produce the antibody, and then there's just the expression purification and shipment to the customer. One area where there can be variability is in conjugation of these antibodies, if these antibodies are used as detection reagents or secondary antibodies. And one of the technological features of the SpyTag-SpyCatcher technology actually confers some benefits in this area of labeling. So one of the things we've done is with some of our catcher formats, we've introduced cysteines, which allow us to do site-specific labeling of these catcher domains. And significantly, this site-specific labeling of the catcher domain, say with peroxidase or with biotin, or even with fluorophores, can occur before the antigen binding fragments, before the Fabs are actually added, so that you can essentially couple your Fabs to pre-labeled catcher domains. And this has a number of benefits. Firstly, the pre-labeling of the catcher domains is something that can be done site-specifically because we've introduced these cysteines, so we can use maleimide chemistry to control, where the label is incorporated. And we have different formats. with either 1 or 3 incorporated cysteines, so the customer can choose the intended degree of labeling. The lot-to-lot reproducibility should be very high when we do this, because we have site-specific labeling, and the customer, we would site-specifically label an entire batch of catcher, and then the customer, say we generate 10 antibodies from them, they could use the Phage of all of those 10 antibodies and couple them all to the same pre-labeled lot of catcher. So there should be no variability in labeling between the different clones. And when the customer's then doing a comparison with those clones, they should be able to attribute any difference they see in performance to the antigen binding characteristics of the antibody rather than the degree of labeling. So if you were to contrast this between using, say, primary amine coupling to couple 10 clones individually, you'd never be 100% sure whether what you were seeing was the antibody binding, the antigen binding characteristics of the antibody, or the degree of labeling between the different clones. Another advantage is that we seem to get higher sensitivity when we have when we use this pre-labeling approach, and we think it's because it means that there's no incorporation of the label into the binding site of the antibody. And I will show you an experiment we did to demonstrate this in the next slide. So this slide shows essentially a comparison of 3 different formats of the same antibody. So in gold, what you see is essentially the Fab antibody subcloned into a mammalian IgG expression vector. A full IgG is made, and then it's labeled using primary amine coupling. And then the two showing the Fab domains in purple are basically SpyCatcher formats. So the Fab is made as a standalone Fab. It is then protein ligated to pre-labeled catcher domains. And what you see in the diagram essentially is the mammalian express domain is shown in blue. So you see that curve is further over to the right. So the analytical sensitivity you achieve with that is about 10-fold less than with the pre-labeled. And as I mentioned in the previous slide, we believe this is because in the SpyCatcher formats, the Fab is added after the catcher is labeled. So we would be confident there's no label incorporated into the paratopes or the binding domains of those Fabs. So all of the Fabs would be capable of binding, whereas what we hypothesize is in the mammalian expressed version, some of the label was incorporated into lysines in the paratope, and so a subpopulation of that antibody will be unable to bind the antigen, which is why you get the reduced sensitivity. Our customers need us to generate validation data for the antibodies they receive from us. This is an area where we're familiar with this kind of work, with the work we've done with IVD companies and with biopharma companies, and it's something we can be flexible and do quite easily. So in this diagram, what you see is 3 production lots of the same clone of antibody, and what you see is very high reproducibility. So if customers need us to produce 3 lots in parallel, we can do this. We can also identify one of those lots as a reference lot. We can aliquot it down and freeze it. And then for every subsequent batch we make of that particular clone of antibody, we can do a comparison study to that original reference lot, so that we can show that the reagent performance is not drifting over time. So we're getting towards the end of the presentation now. I'd just like to ask you to do a quick survey, because it's always interesting for us to know, what are the challenges that you as customers feel are the most pressing when it comes to generating your custom reagents for putting into your bioanalytical assays. So yes, if you could take a few seconds to fill this questionnaire in, it should pop up on your screens, and then I'll go through the last few slides, and that'll hopefully give us time to compile all the data, and then we can speak to this at the end of the presentation. So I'll be silent for about 15 seconds, and then I'll move on to the next slide. In addition to reproducibility, stability is another important consideration when considering critical reagents for bioanalytical assay methods. So in this slide deck, what you essentially have is an accelerated stability study where we're using a 20-degree temperature to stress the antibodies, and essentially what you're seeing is antibodies are very robust, whether they're recombinant or natural to these temperatures. And this is just an example. Accelerated stability is something we offer. We also offer free source stability, and we can also look at the melting point of antibodies as well. That can sometimes be indicative of whether an antibody is likely to be stable or not. So this brings us to the final slide of the presentation. And I hope really the few take-home messages are that don't just think about projects in terms of the specificity that you require. You can engage with your tool antibody supplier or your reagent supplier to consider all aspects of the project. So we can design antibodies for the specificity that will meet your requirements, hopefully. But in addition to that, think about all the formats you may require the antibody in. And once you've finalized which clones you want to take forward, any validation requirements you may have, because what you might find is that we or other suppliers out there may be able to fulfill a number of these requirements. And if it's built in from the early planning of the project, it's something that we can deliver as we work through it. So yes, I think that brings me to the end of the presentation. So let's have a look at the results of the survey, and I'll be happy to take any questions after that.
Thank you so much for that presentation, Paul. I hope our audience found it of great interest and of value. If you do have any questions for Paul, then please continue to submit the questions using the Q&A tool in the right-hand side of your screen. Towards the end of the presentation, we asked you which of these are the biggest challenges you face when generating custom antibodies for bioanalytical assays. So our audience said the top two were timeline for antibody generation is too long and desired specificity is not achieved. So Paul, I'd like to hand over to you to discuss the results.
Yeah, thank you. Thank you, Annie. So I hope in the presentation you saw that both specificity, I spent a lot of time discussing that, but the timeline of phage display is quite a fast process. The actual workflow with no additional extras is 8 weeks, and after 4 weeks you have a reasonable indication of whether your project has been successful and that you know whether you've got hits that can be sort of taken forward for expression and purification. So compared to conventional classical animal immunization based techniques, it is a fast process. Not only is the generation process fast, but the reproduction process is fast as well. So we can offer customers reproduction of E. coli expressed antibodies in 1 to 2 weeks if they opt for our fast track service, or 4 to 6 weeks if they go for our regular service. So speed is certainly something that an in vitro process allows. In terms of specificity, if you have particular specificity requirements, I think the easiest thing is to discuss with us, because as you saw in my presentation, with an in vitro process, there are a very large number of strategies, tactics you can employ to really hone the specificity of the antibodies that you're generating. Yeah, I hope that answered or addressed those 2 points that were raised, but thank you for your feedback. It's always interesting for us to know, to know what the main problems are that customers are encountering when generating their tool antibodies. Annie, I'm happy to take any other questions that have come in.
Yeah, so thank you for those insights, Paul. We'll move on to the Q&A now. So our first question is, have you encountered any differences in performance when switching between formats?
We haven't really experienced differences in performance in terms of affinity or specificity of the antibody. From the presentation, I guess we demonstrated when it comes to labeling, for example, we seem to have higher sensitivity, which we attribute to label not being incorporated into the paratopes of antibodies because of the stepwise method we have for assembling conjugated molecules using the SpyTag technology. In terms of overall binding strength, obviously our antibodies allow you to switch from a monovalent to a bivalent format, which increases the avidity. So you can see sort of predictable differences in binding based on the formats you choose to change to, but we don't sort of see any, I guess, things that would indicate sterical hindrance or sort of problems in terms of actual binding the target specifically when we change between the formats.
Okay, great. Thank you for that. And how many antibodies do you get from a custom project?
Our standard package, we will typically sequence 20 or 30 antibodies, and then the nature of Phage display, the nature of the enrichment is that there can be duplication. So when we sequence 20 to 30 hits, we'll get the sequences and then we deduplicate. So we may expect 10 to 15 unique antibodies, which is incorporated in the price of a project. But there are earlier stages in the project where we use 3, 4 well master plates essentially. So we could get as many as 200 positive hits on those plates, and maybe we have 2 plates per project. So there are potentially 400 hits we could pick and sequence if the customer wants that many, but there would obviously be additional fees for that because only 20 to 30 are included in the project. If we know epitope diversity is hugely important for a customer and they want to look at a very large number of antibodies, it is possible either to pick and sequence a lot more, or we also retain these master plates so if people want to revisit them in a year's time with some new antigen that they want screening against that's relevant either for negative or positive selection, we can actually get those plates out and revisit them. So yeah, again, an in vitro process it can be frozen down. The whole process can be frozen down. It's a very flexible system.
Okay, great. Thank you for that. And can a SpyTag be added to any antibody?
Yes. If we're using our HuCal technology to make the antibodies, any antibody we produce through that process can feature a SpyTag because we just select the appropriate destination vector. If a customer has, say, a mouse hybridoma antibody that they really like and they would like it SpyTagged so that they can benefit from the format change, it's something we can look at doing, but essentially what we would probably have to do is have their hybridoma or their monoclonal antibody sequence, and then we try and produce it in E. coli, and not all mammalian antibodies produce well in E. coli. So we'd make the Fab fragment of their hybridoma in E. coli. If it produces as well, we can make it in the SpyTag format, much like a HuCal antibody. But as I say, not all mammalian proteins express well in E. coli, so we'd have to look really at that if that's something customers require.
Okay, thank you very much for answering our questions today, Paul. Before we wrap up, do you have any last comments you'd like to add?
I'd just like to thank yourselves, the organisers, and thank you to everybody who's attended and listened to the presentation. It's always nice to know that people are interested in what you're doing. So thank you all very much.
Okay, and I would once again like to thank our wonderful presenter, Paul, as well as you, our listeners, for your time and your questions.